And tonight, what I hope to do for everyone is convince you how making major inroads in turning around difficult to treat human disease is really based on understanding the mechanisms of disease, the underlying molecular and cellular biology of the disease. Using the example of melanoma, a disease I've been privileged to work in now for a number of years, as the example, but the message to me is clear. If we want to overcome difficult disease, we must focus on understanding the fundamental mechanisms of the disease. And tonight we'll talk about melanoma, but haven't we seen that with COVID, some remarkable achievements in controlling that disease through science. So I'll start off with my disclosures, and I have to acknowledge that David's recording me, and so I've done that, and now just bear with me while I get my things working. There we go. Now we're advancing the slide. So I have some disclosures in that my institution, the Peter Max, received some funding from industry as listed here for clinical trials that I have led. So what I'll do for you tonight is I'll talk about the disease of melanoma in Australia and New Zealand and show you why it's Australia and New Zealand's disease. A bit about understanding the biology of melanoma and some of the fundamental discoveries in our thinking about cancer and melanoma. Then, to me, what is just amazing and, you know, gets me out of bed every morning, and that is taking that knowledge of biology and turning it into better outcomes for patients. And I'll walk you through, you know, what happened with melanoma. Then really focus on, you know, the outcomes of all of that translation of science into treatment. And that is, you know, the remarkable situation now when I sit down with a patient with melanoma who has advanced disease that has spread around their body, and I tell them we are aiming for cure. And that's a pretty nice thing to be able to do as a clinician. And then talk a little bit about the future and how we're going to keep continuing to improve this disease.
I'll just interrupt you there to say that if people listening have any questions, then feel free to put them in the chat box, and then we'll put them to Professor McArthur at the end of the talk.
Thanks, David. Okay, so we'll start off with the impact of the disease of melanoma. So first of all, what is melanoma? So basically it's uncontrolled growth, malignant transformation of the pigment cells in our skin called melanocytes. So in the base of our skin, we have these pigment producing cells. Mine in my scalp don't work very well, which is why I have grey hair. And in acknowledgement of our chair tonight, indeed, the molecule that Professor Vo discovered, BCL2, is lacking in my hair follicles, which is why I have grey hair. And that's because the melanocytes that produce the pigment when you have grey hair undergo cell death and no longer produce the pigment. But when those cells get malignantly transformed, we have the disease of melanoma. And shown on this slide, the nasty nature of that is when it starts to invade below the skin and can travel through lymphatic vessels or blood vessels around the body. So melanoma of the skin, Australia is shown in that red bar graph, and you'll notice another big bar graph there, which is New Zealand, have the highest rate. So this is the cases per 100,000 of population, which is age standardized, the epidemiologists like to do per year, and Australia and New Zealand are way out ahead of the pack. You'll see the next most common countries are European countries, Germany, Sweden, the Netherlands, where there's very low rates in countries north of us in Japan and Korea. And so it is a disease of people of European origin. And as I'll get to Australia and New Zealand, we have very high ultraviolet light exposure in our countries, which is why we have the highest rates in the world. Now, the graph I show you on the right is that with time, the rates of melanoma are increasing. And I won't go into the reasons why, but we can pick that up in discussion if you're interested. The orange graph there is the mortality. You can see it's been pretty flat. But what I'm going to show you later in the talk is that that's changing dramatically now, and it's changing because of the fact that we understood the biology of the disease to come up with new treatments. So Australia, New Zealand's disease, curable if you catch it early. So if you can remove it surgically, you get good outcomes. But it is the third most common cancer in Australia. Someone is diagnosed every 30 minutes in Australia. Sadly, someone dies every 5 hours. And it is the most common cancer in you might say people at the prime of their life. You know, David and I might think we're at the prime of our life, but the 15 to 39-year-olds really are. And that's when we did our best science, David. And unfortunately, it's the most common cancer in Australia and New Zealand in that age group. Now, you'll see on the left there a cartoon of a sun. So 95% of this disease is caused by ultraviolet radiation, which damages DNA, leads to single nucleotide variants, point mutations in genes, and that drives the carcinogenesis, which I'll talk about a little bit later. So we have a staging system for all cancers. We have one for melanoma. The one we use is called the TNM system. So T for tumour, N for lymph nodes, because it can spread to lymph nodes, M for metastases, which is a distant spread around the body. What's actually rather nice about melanoma is that we have an international collaboration where we put data together from over 46,000 patients and define our stages according to outcomes from real data, rather than a consensus, which happens in a number of cancer in the staging system. So the data in melanoma, and when I used to give lectures more than a decade ago, I didn't have many treatments to talk about. I talked about what a wonderful staging system we have. But now, as you'll see tonight, we've got a lot more. So stage 1 is localized to where the tumours start. Stage 2 are tumours that are of a larger size. Stage 3, when it starts to spread to regional areas of the body, most commonly lymph nodes. And stage 4, which I'll talk about a fair bit tonight, is where it has unfortunately spread around the body. So it's that stage 4 disease that had average life expectancy of 9 months until the science that I'll talk about tonight came to bear. So we'll be showing you a lot about stage 4 disease tonight. All right, so you can't overcome a tough disease unless you understand the biology, and that requires discovery science and original thinking. And so I'll just walk you through now a bit about understanding the biology of melanoma. So I take these images from a review from 3 immunologists published a while ago. But what you'll see up the top here is that normal cells, when exposed to carcinogens, in the case of melanoma, that's ultraviolet light, undergo various genetic damage to generate drivers of the malignant phenotype. I'm not going to go into the details, but the thing I want to point out on this slide, which has turned out to be highly relevant, is that melanoma is close to top of the pops for having the most mutated proteins in its genome. So in melanoma, there can be up to 100,000 mutations, point mutations occurring in genes that encode proteins in an individual tumour, huge numbers. What that does is it generates a lot of so-called neoantigens. So these are tumour antigens that are novel and due to mutated proteins that the immune system can recognise and respond to. And that is absolutely key for some of the treatments I'm going to talk about this evening. Now, quite a number of years ago, it was in fact Ehrlich and later Macfarlane Burnett, famous Melburnian scientist, came up with the idea of immunosurveillance. Maybe our immune system, controls and prevents us from getting cancer. So this gentleman, Rob Schreiber, came up with the idea of cancer immunoediting and the real role of a molecule, gamma interferon, as a key molecule in controlling the emergence of cancer using beautiful fundamental science in mouse models. Came up with a 3-stage idea of the development of cancer, where the immune system fails. That initially the immune system is successful, it eliminates cancer cells, prevents us from getting cancer. If there are changes in either the immune system or in the tumour cells, that they can start to no longer be controlled, and there's a state of equilibrium. And then in escape, the immune system can no longer control the tumours. So the yellow cells you see here, very important in the equilibrium phase shown in these beautiful mouse models, but also in the escape phase, CD8 T cells. And T cells, of course, have a very famous Australian history as well, because Jacques Miller discovered the role of the thymus, the organ that produces T cells, and put this alternate immune cell T cell on the map. And I have given lectures with Jacques in the audience and paid homage to what he's done for patients and what I'll talk about tonight, because that discovery of the T cell, nothing would have happened without that. And Schreiber, shown here, really proved in mouse models that this immune surveillance really did occur and that the immune system was important in cancer. So what about melanoma? I've already told you there's lots of mutated proteins. Well, in fact, we've known for a long time, shown on the right here, is what a pathologist sees down the microscope when they look at a melanoma, is that those little blue dots are lymphocytes, including T cells. And melanomas contain those. And in fact, if you've got a lot of them, there's a lower chance of relapse compared to if you don't have them. So this was a clue that immune cells and T cells are important in melanoma. And then for many years, we were trying to harness that with a variety of different interventions. So I just show on the left here a molecule called interleukin-21, which we put into a clinical trial a number of years ago. And what this graph is, we use this a lot in oncology, it's called a waterfall plot. And you'll see that is measuring the dimensions of tumours before and after treatment. So on the right of that graph, you can see that, apologies, I'll just go back. You can see that there's one patient here whose tumours, had a waterfall. They dramatically reduced by 70%. That's a so-called partial response. But unfortunately, with this immune treatment, most patients' tumours actually grew. And so, my American colleague, Tony Rebas, often says, well, in melanoma, we had geyser plots because the water was going upwards rather than downwards. And I'll show you how that has changed dramatically into true waterfalls. But what this data shows us is occasionally patients responded to our attempts at activating the immune system. And that was important because it kept our interest up, and I'll get to later the fundamental discoveries that turned everything around. Now, the other important biology in melanoma is mutated genes that activate so-called oncogenes, genes that will drive normal cells to become cancer cells. So my colleague Mike Stratton and his colleagues at the Sanger Centre in the UK had completed sequencing the human genome, rather small experiment. And they had a lot of sequencing machines, DNA sequencing machines, looking for things to do. So they started sequencing cancers. Let's look to see if there are, mutated genes to be discovered in cancer. And they discovered a protein kinase called BRAF. And this is the data from their initial paper back in 2002. And actually, they found that 45% of melanomas had mutations in BRAF. So this paper really caught my attention. And then walked in the door of my lab a young aspiring dermatologist called Wendy Liu. And Wendy said to me, I want to do a PhD with you, Grant, and I want to do it on melanoma. And actually, I wasn't working on melanoma at the time, but I said, gee, you know, Mike Stratton colleagues just found this very interesting mutation in BRAF. Wendy, why don't you work on that and develop a test so that we can find this mutation in patients and study the biology of the BRAF mutated melanoma? And so that's what Wendy did for her PhD, but what was very successful was establishing a test so that we could find patients with a mutation to put on clinical trials that I'll get to. So what is this BRAF? So it's a protein kinase. It is an enzyme that leads to phosphorylation of other proteins. So in normal cells, BRAF is fairly quiet. It forms protein dimers with its related protein CRAF. And there's a feedback mechanism, which we see a lot in signaling in cells, that dampen down the signals to cell growth. However, in melanoma, when this gets mutated due to the UV light, the BRAF no longer forms dimers, it functions as monomers, and it's hyperactive with over 100 times its normal capacity to function to phosphorylate other proteins, and that's what drives the proliferation of melanoma cells. So what all this discovery science led us to was the idea that we have a disease that is an oncogene-driven cancer by BRAF, shown in the melanoma cell on the left there, and then there was the hypothesis related to a bit of success from stimulating the immune system, lots of T cells which were associated with good outcome if you had a lot of them in melanomas. And so maybe, you know, melanoma was an immunogenic cancer. We'd found this oncogene. So, you know, could we use that to help patients? And so now I'll get into what is, you know, I think, you know, to me, something that was very exciting for me to be involved in my career is turning that biology into treatments for patients. So the first treatment was a drug that turned off this enzyme called BRAF, discovered through beautiful science in a small biotechnology company in California called Plexicon. And what they did was they took the BRAF protein, made crystal structures of it with and without the mutation, and then did a lot of structures binding different small molecules to determine the best structure or shape of a small molecule that might be able to turn off this enzyme activity BRAF. So they found a very selective compound. The little table there on the right shows you that this particular molecule they discovered called vemurafenib, potent inhibitor of cells that had the BRAF mutation, but if you didn't have the BRAF mutation, the drug did not work. And just shown at the bottom left there is the crystal structure of this molecule binding to mutant BRAF with high selectivity. Now on the bottom right there, and I'll just blow it up for you, is what really caught my attention, which was a bit unusual in cancer, and that is, oh, my slides are stalling again. Here we go. Just to blow this up for you here, and that is that as you increase the dose in animals that were growing this BRAF mutant tumor, you went from slowing the tumor down to then actually killing it and getting regressions of tumor that was totally dose dependent. And so, you know, it was unusual to have such exquisite sensitivity that was dose related in shrinking tumors with a drug in animals. But that caught my attention as being likely to be a highly selective drug that would work in humans. So based on Wendy, the PhD student who developed the test, we were able to find patients that had this BRAF mutation, enroll them on clinical trials. This is the first of our now true waterfall plots. You only see one geyser there, mostly waterfalls, because virtually every patient we treated had shrinkage of their tumour, quite remarkable. Now, other scientific data that came out from that initial phase 1 trial, the first stage of trials you evaluate new cancer drugs in, we're able to look at the molecules that BRAF talk to. It's a protein kinase, so it phosphorylates other proteins. So we could look at molecules downstream and see whether they were getting phosphorylated or not by this enzyme. And what was remarkable was that the more we're able to inhibit this phosphorylation, the more the tumours shrank, is what this graph shows. So, you know, showing really we're on track, and it was mechanism based. We were turning off the proteins we should have been turning off, and that correlated exactly with whether or not a patient's tumour was going to shrink. Then we had to, of course, to make the regulators happy that this new treatment was worthwhile. You have to do a randomised trial to prove that beyond doubt that it is working. So patients that had advanced melanoma, that had metastasized, spread around the body with this BRAF mutation, underwent a randomised trial with the new drug versus the previous chemotherapy that didn't work very well, called dacarbazine. As I said, 9 months average survival for these patients. And so this first trial, these were the results. Definite improvement in overall survival. And this is a survival curve. I'll show you a couple of these tonight. So on the y-axis there, percentage of patients alive, time is on the x-axis. So with time, some patients sadly die, but a lot less deaths happening in the patients that got the new BRAF inhibitor. Now from there, you know, we wanted to get better because there were still patients dying, and we knew that BRAF worked through turning on another protein called MEK, and drugs were being designed there. So we figured that putting the two together would more effectively turn off all of this biochemistry in the melanoma cell. And now I'm going to show you another one of these waterfall plots. And this one's even more impressive. So if you remember the last one, first of all, every single patient on this trial of putting the two drugs together got shrinkage. And the depth of the shrinkage was a lot more as well when you put the two drugs together. And so in cancer, we find that combination therapies usually work better than single drugs. And this turned off the fundamental biochemical pathway that was making the cells grow more effectively. Now that's the BRAF, the mutated oncogene. I'm going to swing over now to talk about the discovery science for new immune therapies with so-called immune checkpoints. Now, I show some key people here that made some discoveries. So the top two gentlemen, Jim Allison and Tasako Honjo, who received the Nobel Prize for their discovery of the concept of immune checkpoints. But underneath that, there's, you know, often Nobel Prizes, you know, it's a maximum of 3, and it's to those people that clearly made a huge impact in physiology and medicine. However, often there are many others that made very important discoveries. So underneath Jim and Honjo there, Gordon Freeman, Eileen Sharp and Li-Ping Cheng, who really did a lot of very important work here as well that empowered the clinical development of these immune checkpoint therapies. Now, just getting back to my favourite topic this evening of discovery science starting it all, I tweeted the day that Jim won the Nobel Prize as follows. There's my quote, but read Jim's, what Jim's quote was when he got when he got interviewed and put out on Twitter the day he won the Nobel Prize. He said, "The reason I'm thrilled about this is I'm a basic scientist. I didn't get into these studies to cure cancer, even though, as we see, he did cure cancer. I wanted to know how T cells work." And so, you know, I love that quote from Jim, and I think it just says everything about the importance of discovery science in overcoming difficult diseases. So, so what Jim discovered was a molecule called CTLA-4. This was a brake on T cells. So T cells get presented with antigens, whether it be an infectious disease like COVID or whether it be one of these mutated neoantigens on a melanoma cell. And then the T cell will respond and it will proliferate, but it has brakes. And one of these is CTLA-4. If you turn that off, then you get more T cells responding to the antigen. Now, those T cells, I've already shown you how they can be inside a melanoma. But melanomas and other cancers, you know, pretty clever. It's really Darwinian evolution. They upregulated molecules that can effectively switch off the T cells. So that's where you get Schreiber's immune escape. That these a molecule that Tasako Honjo discovered called PD1, program death 1. The kids love that because it sounds very science fiction-like, program death 1. But it's a negative break on T cells. And when the tumour cells push that break, the T cells stop proliferating. And so that's PD1. So these were the two immune checkpoints in T cells they won the Nobel Prize for, and as I'll show you now, were absolutely critical in developing new treatments. So there were antibodies to turn off the function of those molecules. And, you know, I always like to remind people that here without Australian science, we wouldn't have these antibody treatments because, first of all, it was Henry Harris at Melbourne University that identified that you could fuse cells together, and that led to the monoclonal antibody technology down the line, where you could take a B cell that produces an antibody and an immortalized cell and make an immortalized cell that can produce large quantities of antibodies, and that's what these therapies are. So that was required, but also required was a discovery from Gus Nossel that actually one B cell makes one antibody. And those fundamental science discoveries led us to these monoclonal antibody technologies that now I'm going to show you have had such wonderful outcomes for melanoma patients. So this was another waterfall plot. I love showing you waterfall plots, this time anti-PD1, the immune checkpoint molecule in patients with very advanced melanoma. And look, yeah, there's mostly waterfalls here and not many geysers. In fact, 72% of patients had some tumor shrinkage. So for an immune treatment, remember that graph I showed you very early, where there was one patient that got substantial reduction. I mean, this was hugely different. Indeed, my colleague, Rick Kefford, who was involved in these very first trials in Sydney, you know, was quoted as saying, "This stuff's penicillin for cancer," which is a big statement, but it was exciting times and it really has benefited many patients, as I'll get to more. So this particular trial just kept enrolling more and more patients on a phase 1 trial, first time in human clinical trial, another one of these survival curves, which is pretty good for very advanced melanoma. And so actually out of this phase 1 trial, the FDA and then actually the Australian regulators approved a cancer treatment that hadn't even gone to phase 2 or 3 clinical trial because the results were stunning. Penicillin for cancer, as Rick said. So the point of this part of the talk is that it was that fundamental discovery science that led rationally to these treatments that have had a profound impact. And now I just want to switch over to talking to you about, you know, this idea of cure, and show you the data and try to convince you that, in fact, our patients with very advanced melanoma, some, but not all, can be cured. So, so where we're at now is we have predominantly combination treatments. We turn off the BRAF oncogene and the related molecule downstream, and we also can turn off both of the checkpoint molecules discovered by Jim Allison and Tasako Honjo in CTLA-4 and PD-1 in combination as well. So to show you now the longer term outcomes for these patients that we published last year, presented at a meeting by my colleague Jed Walchop from New York. So these again, more survival curves where patients, whether they're alive or not. And the red graph there is the combination of CTLA-4 and PD-1. We're now out at 6.5 years, around 50% of patients are alive, when the previous treatment I mentioned, you know, historically was average survival was only 9 months. So we also analysed, for those that like lots of detail, melanoma specific survival, because actually if you're curing people of disease, you know, they will die of something else. And so we wanted to really look at, because people would die of other things, after you got rid of their melanoma. So in fact, 56% of patients out at 6.5 years were alive from free of melanoma that had not died of something else. The reason, 49% versus 56% is 7% have died of something else. So we're now starting to look at melanoma-specific survival.
Grant, what stage were these patients at to begin with?
So these were all advanced. So they were all patients spread around the body, stage 4, with, historical controls, average survival of 9 months. And now you're talking, over 50% alive at 6.5 years. Okay, so the targeted therapy with the BRAF doesn't seem to do as well, and I'm not going to show you. We've actually done comparisons now, and the immune treatments do better. But still, there was around just over 30% of patients that are alive at 5 years and free of disease with the BRAF targeted treatments as well. So getting long-term control. So are we really curing these patients, which is cure is actually a hard question for a hard word to use for oncologists. Some don't like it. They're used to, unfortunately, patients with advanced cancer passing on, and are very conservative about using the word cure. So what do we really mean by cure of cancer? Well, no evidence of disease, so you can't find, tumor lumps on scans, not on treatment. If you're cured, you no longer need treatment. And a normal life expectancy. And I started touching on that with this melanoma-specific survival concept. So, that's hard to say to someone, oh, you've now got a normal life expectancy. It's a hard call to make. So a bit of a surrogate of that. We think now if you get to 5 years, no evidence of disease, not on treatment now with melanoma, we believe these patients are cured. Now I want to show you that's not just my view, because what I've done when I've lectured around the world on this, I've surveyed live all the melanoma experts in the room. Do you think we cure people? These were the answers when I polled a room of 200 experts a couple of years back in Europe. And 58% says, yes, we're curing patients with both the immune treatments and the targeted treatments. Only 20% were skeptical and think that we weren't curing people. Now, I've also asked a group of patient advocates and experts by experience that have an interest in melanoma, exactly the same questions. They're actually a bit more optimistic. 73% of the patient advocates thought we're curing people with both the immune treatments and the targeted treatments. Okay, so what's the impact of all of this, in a societal point of view across population of our population? So we're lucky here in Australia to have fantastic cancer registries. In fact, the first cancer registry annually that reports its results is the Victorian Cancer Registry. And so we have very, very updated data. And this shows an epidemiological endpoint. So this is age standardized mortality from melanoma per 100,000 population. So it's adjusted for age. And you can see that a couple of things here. Firstly, men do worse than women. They're more likely to get melanoma, and in fact, the prognosis is a bit worse in men as well. And that, between 1980, which was, David, around the time you and I started medical school, or close to it, through to 2010, so 30 years, no real change in the mortality from this disease. But have a look between 2013, after these new treatments got approved, now to 2020. There's been a sharp reduction in the mortality from melanoma in Victoria. In fact, it's a 50% reduction in age standardized mortality in that period of time. So it's not just clinical trial patients that go onto the trials that are getting all this benefit. Out in the real world, we've actually slashed the mortality from this disease in our society. So very significant impact. Okay, so now just the future. What is the future of this disease? I'll run you through a few concepts that we're, really thinking about and starting to apply now. Now, first of all, prevention is better than cure. Melanoma is a disease. We know the carcinogen. It's UV radiation, either most commonly from the sun. On the right there, we've now pleasingly banned solariums in this country. Other countries still use them, but you can prevent this disease because we know the carcinogen. So we should keep doing that. The slip, slop, slap, etc. need to be applied. Prevention is definitely better than cure. The other interesting thing about melanoma of the skin is that, the skin is accessible for examination. And so we can detect melanoma early. And I mentioned to you that 90% of melanomas are cured with surgery alone. So if you detect it early, it's much simpler than having to go on to the treatments I've spoken about. I haven't talked at all tonight about toxicities, but the treatments do have some toxicities. And so much better to detect early, a simple operation to cure. This shows a dermatologist using a device called a dermatoscope, which allows for greater detail of examination of lesions on the skin and more accurate diagnosis of whether something is melanoma or not. And Australia has led the world here in showing that one can train GPs to very accurately diagnose melanoma using this technique. And so you can detect the disease early simply with cure. A very interesting technology emerging now, and there's a national trial running in Australia to evaluate this, is total body photography. This particular device, the Vectra machine, very quickly enables someone to stand in there and then get all of their skin photographed, and in very high resolution that can then be examined by a skilled operator to determine if any of the lesions on the skin are changing. And what is very innovative there is that's being linked to artificial intelligence to see if one can teach a computer to make the diagnosis of a melanoma and detect it early. So prevention, early detection of this disease remained important. But what about the next stage of development of these new treatments? So one perhaps obvious question is, we've got an immunotherapy where actually we're turning the T cells in patients' T cells or non-malignant cells on to eradicate the tumour. And then we've got targeted therapy where we're using this targeting BRAF in the tumour cells. So what if we put the two together? Could you get better outcomes if you put the two together? So we now have done trials of that. And in patients with, again, advanced, that stage 4 melanoma I've been talking about all night. And yes, you do get better results if you add an immune treatment to that, to the targeted therapy with BRAF. But, it's not a home run. This is not preventing, death, taking, upwards of 50% of survivors, well above, up into the 80 to 90% or above where we'd like to be. So it's better, but it's not a home run. And so studies are underway to determine, can we pick the patients that may actually benefit from putting these two treatments together. Now, the area that, I'm pretty optimistic about as an approach to continue to improve the survival of patients with this disease are based on, again, fundamental biology happening in laboratories. And that is that it is clear that the amount of cancer in the body, in this case melanoma, is related to how the immune system functions. So if you've got a high burden of cancer, that can suppress the immune system. And so colleague John Werry and colleagues, a tumour immunologist in the US, published a few years ago in mouse models some very nice data to show that if your T cells can proliferate and there's a low burden of disease, and those two things are related, then in mice and validated in patients as well, the data I show you here, that you get much better survival if you've got low burden of disease and T cells that can proliferate that are associated with low burden of disease. And what's rather nice, again, mechanistically understanding the biology, is that one mechanism this occurs is that there's little fatty droplets of tumour cells that can float around the body called exosomes. And they actually have in the exosomes the ligand or the molecule that binds to that PD-1 I spoke about and can turn off T cells. So the more melanoma you have, the more exosomes you have that have this PD-L1 molecule that turns off the immune system, which is a mechanism by which the burden of cancer can be related to turning off the immune system. So what does that mean? It means that we should try to treat and detect cancer early when it's in low burden of disease, and then we'll get better results with these drugs. And so the way we do that is by so-called adjuvant, and also another approach called neoadjuvant immunotherapy. And this is a slide provided by my colleague Christian Blank from the Netherlands, cartoon form to explain what we're trying to achieve by giving drug treatments, in this case immunotherapy, in so-called adjuvant or neoadjuvant setting. So the first across the first row there is the rationale for adjuvant therapy. So a surgeon removes a melanoma, but there will be some cells that are already spread or left behind. We can come in with our immune treatments and try to activate the T cells to eradicate those original cells, and that will occur. But that, perhaps we haven't got an optimal number of T cells able to recognise the tumour cells, and that's why some patients, even despite having this adjuvant immunotherapy, PD-1 antibodies, the adjuvant immunotherapy can still relapse. So an alternative approach is so-called neoadjuvant approach, and that is that you give those antibody treatments while the tumour is still in place, before the surgeon has removed it. And the hypothesis is that maybe you will get more and more diverse T cells activated, such that after the surgeon removes it, any cells remaining behind are more effectively controlled by the immune system. So that's the hypothesis. We're still on the path of testing that hypothesis, but I'll show you some of the early data that is pretty promising and exciting. So first of all, Christian and his colleagues at the Netherlands Cancer Institute did a small trial where they compared adjuvant, so where the treatment comes after the surgeons remove the disease, to the neoadjuvant, giving the immune treatment before the surgeons remove the disease. And what they looked at is the number of different clones of T cells, and also whether they were expanding following the immune treatments. And what they showed was that the neoadjuvant, you got more T cell clones that expanded more than you did with the adjuvant therapy. So promising biological data. Now we know that adjuvant therapy works well. So both the immunotherapy on the left, compared to placebo, much better outcomes, survival curves, again, like I've shown you before. The blue is the immune treatment, the red is a placebo, or indeed for target therapy with the BRAF drugs that I've spoken about, much better outcomes. But now we're starting to get data of this neoadjuvant approach, where the tumour's still in place when you start the drugs before surgery. So if you're in the group of just over 50% of patients that get a complete pathologic response, basically there's virtually no relapses occurring in melanoma when you give immune therapy. Target therapy with the BRAF doesn't look quite as good, but still pretty good results if you get that pathologic complete response. That is when the pathologist looks after the surgeon removes the tumour, there are no viable cells there. I mean, that is data that is, that I think is very, very promising indeed. And so we're doing a trial now of adjuvant versus neoadjuvant. There's actually two trials underway, one a collaboration between Australia and the Netherlands as the two lead countries, and then an American trial as well that are going to formally test that. So the science is telling us it should work better, and now we'll see from the trials. All right, so I'm going to stop there, David, and delighted to take any questions. I do want to acknowledge a few people. So just under the word acknowledgements there, Wendy Liu, young PhD student, walked in the door and set up all the testing so that we could get our BRAF trials going. And then on the right, there are a whole bunch of people that I've worked with on these clinical trials. You've got to very much acknowledge the patients and families and their supporters that really donated specimens to study the biology of melanoma before we had treatments, and then got involved on the trials. And this is just my team around the time that the BRAF drugs got approved that worked on this. And the point I want to make is this is a bunch of molecular biologists, pathologists, imaging people, research nurses that collected the data and cared for the patients and clinicians. And that's what you had to bring together to sort of make a big difference with these, with these treatments. So David, I'll stop there and happy to take some questions.
Well, that was absolutely wonderful, Grant. And I'll take chairman's privilege and ask the first couple of questions. So with the checkpoint inhibitors, the immune modulatory drugs, do you ever combine the two of them? Do you combine a PD-1 inhibitor with a CTLA-4 inhibitor?
Yeah, and indeed, yeah, we get better outcomes if we do that. And the simple mechanism by which that occurs is that the CTLA-4 really expands the number of tumor responsive T cells, but they're not fully switched on, where the PD-1 comes in and then switches on those expanded T cells, so you get some synergy together. One of the other things that is still hypothesis and we need more follow-up is that, the unique thing about the immune system, of course, is memory. We have our COVID vaccines and months later, we're still making antibodies because our B cells have memory. And possibly the CTLA-4 PD-1 combination will generate more memory cells than either one alone would. And so, that's still to be determined, but that's what we're hopeful, that we'll get strong activation of immune memory and then hopefully longer disease control and more cures.
And again, I'll encourage people to put their questions in the chat box. There's a question here from Hamza Pathalakath. Is there any role for TLR7 agonist imiquimod, how do you say that, in treating melanoma together with checkpoint inhibitors?
Yeah, so first of all, David, you pronounced the imiquimod very accurately. So,
very slowly.
So this is a small molecule that is an agonist for a receptor called the toll-like receptor 7. And this is a very strong signal that generates an emergency inflammatory signal and is quite
adjuvant-like activity.
Yeah, is quite immune stimulatory. In fact, the other type of skin cancer, which metastasizes a lot less rarely than melanoma, basal cell carcinoma, that's actually the most common cancer in the world because, a lot of us, particularly in Australia and New Zealand, are going to get them in our lifetime, very common tumours. Anyway, you can put imiquimod onto basal cell carcinomas with a very high percentage of cure, actually, by this toll-like receptor 7, which activates a strong immune inflammatory response. So yeah, it's a great question because those studies have been done. There's a variety of different toll-like receptors, and a number of different molecules have been developed that are being put together with the immune checkpoint inhibitors in current clinical trials. Probably the most challenging thing there, which is practically, is that they're going to work best if those agonists are in the tumor. And if you've got either metastatic melanoma with melanomas metastasized around many organs of the body, or even in that adjuvant setting where you've got micrometastases in patients, you really need to get those agonists to the tumours to elicit the immune response. And so they can work very well when you put it on the skin, but it's getting to the liver, getting to the brain, you know, that is the challenge. And unfortunately, if you deliver them systemically, they do have quite a bit of toxicity, and perhaps you don't get the same concentrations you need to get an optimal immune response. So jury's still out, but it's studies are underway, and I suspect they'll have a role for some patients. You know, I have a just anecdotally a patient who had initially got some response to the immune checkpoint inhibitors. They had a lot of tumours on their skin. And so we put one of these toll-like receptor agonists on the skin to try to activate things on top of the immune checkpoint drugs. And unfortunately, tumours started to grow. And so I referred this patient off for a phase 1 clinical trial to one of my colleagues. They did a scan and, oh, hang on, all the tumours have gone away, because actually was a delayed response. So in fact, the toll-like receptor agonist did work, but it was delayed with the immune checkpoint inhibitors. And pleasingly, that patient's been in remission now for a couple of years. So
you mentioned briefly that there are some side effects from the immune checkpoint inhibitors. Can you tell us a little bit about them?
Yeah, so one could give a whole nother talk on that, because it's fascinating area. So the side effects are due to immune activation. So if we go back to fundamental science, and if you delete CTLA4 or you delete PD-1 in mice, they get inflammatory autoimmune disease. Okay, so they're the type of side effects that one also sees in patients. You get sometimes classic autoimmune disease, other times inflammatory disorders. So the most common inflammatory disorders and immune diseases are involving skin inflammation, so skin rashes, liver inflammation, so we get hepatitis type syndromes. We also get, and this is perhaps one of the most interesting, is endocrine dysfunction due to inflammation, autoimmunity in endocrine organs. So this is the thyroid, pituitary, adrenal gland. Even classical type 1 diabetes can occur in a small subset of patients. There's something about you know, the ability of these molecules, treatment molecules, to turn on inflammation and immunity that can target the endocrine organs. So all that can occur. Pulmonary inflammation is also inflammation of lungs can occur as well. In fact, I say to my patients, any organ of the body can get an inflammatory or autoimmune disorder.
And then if you reduce the treatment or stop it, then
yeah. So reducing doesn't work as once the inflammation has begun. What does work is immune suppressive drugs. So corticosteroids work well, and a whole variety of antibodies to pro-inflammatory molecules like tumor necrosis factor, for example, can work very well, particularly with bowel inflammation is a particularly problematic one. Now, you know, that all sounds pretty awful, but you know, I say to people, these are the safest drugs I've used in my career, because only 10% of patients that get PD-1 will get a significant side effect that requires, you know, substantial intervention, which is pretty good. When we put the two drugs together, David, you asked about CTLA4 and PD-1, that rate goes up to 50%. And what is fascinating, and you know, we don't really understand the mechanism here, that if you get one of those immune side effects, you're more likely to get benefit for your melanoma, and you can turn off the immune system, control the side effects, but the T cells continue to control the melanoma.
Oh, fantastic.
And that is the mechanism we don't understand, and we need to, because it appears that one can, you know, control the adverse effects of activating T cells while maintaining the benefit against the tumour. So why that is the case, we need to understand.
Now, I saw recently in the New York Times, they talked about treatment of very early rectal cancers, I think they were, ones that had a particular mutation that increases the number of mutations in the tumours. And they said every patient responded really well. But, you know, up until then, these checkpoint inhibitors didn't seem to be particularly good for colorectal cancer. Have you got any comments about that?
Yeah, no, that's a fascinating study. So that was just presented at the American Society of Clinical Oncology meeting in Chicago 2 weekends ago. And so what is really interesting here is getting back to this concept that of neoantigens and having a lot of mutated proteins on cancer cells that are recognized by T cells. So this study was in patients that had microsatellite unstable rectal cancers. Okay, so these were patients that had lost one of the genetic stabilizers in their tumor cells. There's a series of proteins that control genomic stability, that if lost, you get a lot of mutations and so-called microsatellite instability, which can be measured, you know, with genomic DNA testing. So all of those patients had that particular mutation, and they're very sensitive tumours to these immune checkpoint drugs. So what this study did was in rectal cancer was to put chemotherapy together, which is the standard for treating rectal cancer, with the immune checkpoint therapy, and got this amazing 100% response rate. So what we need to understand, though, is the other 90% of patients who have microsatellite stable disease and much lower mutation burdens in colorectal cancer, and work out how to activate T cells there, because we haven't got there yet, because they don't respond well to these immune checkpoint inhibitors.
So I've got now a statistical question. Australian life tables would show that an expectation of 6.5 years occurs when you're aged about 85, right? So if you're 85, would it be that for a patient of that age, that 6.5 years cancer-free would be fair to claim a cure, but for younger patients, you'd have to show longer survival?
Yeah, like, that would be one statistical argument for cure, you know, and is to use the life tables as you suggest. But of course, you know, you can get other diseases, and you can be hit by a bus as well. And so, you know, hence why we do have these surrogates, you know, like, so hence why we, you know, if you've got no disease, you can see on scans you're off treatment and you get to 5 years, we're using that as a surrogate. That would be one way of defining it. It's, you know, what is interesting is another thing, since you've mentioned older individuals, that interestingly, when you age, your immune system becomes less effective. There's a phenomenon called immune senescence. And what's interesting, though, is that there's not much drop off at all in the ability of these drugs to work as you get older. So because these drugs are safe, I mentioned only 10% get serious side effects, we do treat 85-year-olds. In fact, I just saw a guy yesterday with a different cancer, a different type of skin cancer called Merkel cell carcinoma, who's aged 92, and I'm treating him with one of these drugs because they're safe. And
well, Jimmy Carter.
And Jimmy Carter, the American president, absolutely. He had metastatic melanoma. It actually spread to his brain, and received the PD-1 drugs and got into remission, you know, at a ripe old age, and you know, tolerated the treatment very well. The other interesting thing there with Jimmy Carter is you know, we used to think that our drug treatments for cancer don't work for brain metastases. In fact, it was just the brain metastases is just a very aggressive and difficult site. In fact, they work just as well. And these immune checkpoint inhibitors, we've shown very good outcomes in patients with brain disease.
You don't have to put it in directly into the CSF.
No, that's right. In fact, fact, if you can see lumps on a scan, you know, the T cells or the BRAF drugs will get into the tumours just fine. You don't have to give it into the cerebrospinal fluid directly into the brain.
Well, I think we should wrap it up there and just thank you for a really spectacular talk.